I want you to picture your gut for a second. And most of us just imagine this simple digestive tube, right? Like a basic plumbing system.
Right, just a pipe that moves food along.
Exactly. But I want you to completely reimagine it. Think of it as a highly fortified microscopic border wall.
Oh, that's a much better way to look at it.
Because on one side of this wall, you have the completely sterile, highly secure internal sanctuary of your immune system. And then on the other side...
Which is just fractions of a millimeter away. I mean, it's so close.
Yeah. You have this chaotic outside world, trillions of living bacteria just passing through every single day. So what happens when that border wall breaks down? You know, when the guards start firing at the crowd and the battle just never stops.
It's a terrifying reality for millions of people. When that delicate truce collapses, you're looking at the landscape of inflammatory bowel disease, or IBD.
And our mission for this deep dive is to unpack the pathophysiology of IBD using the compiled sources. We're going to explore exactly how this chronic gut inflammation causes disease, focusing heavily on its two main poles, which are Crohn's disease and ulcerative colitis.
Right, the big two.
We'll cover the surprising origins of their names, the core biological breakdowns that cause them, some genuinely baffling medical paradoxes, and why this is rapidly becoming a massive global health issue.
It's really a fascinating puzzle.
Okay, let's unpack this, because before we get into how the wall breaks, we really need to map the territories. And we have to clear up a massive public misconception right out of the gate.
Yes, please. The name game in gastroenterology is a huge hurdle for patients. People hear similar terms and they just, conflate them.
We have to state this clearly. IBD is not IBS. Inflammatory bowel disease is completely different from irritable bowel syndrome.
Worlds apart, even though the acronyms are practically twins.
Right. IBS is a functional disorder. And I mean, it is very real. It is debilitating. But if you look at the gut tissue of someone with IBS under a microscope, there's no visible damage.
Exactly. The plumbing looks normal. It's just not working correctly.
But IBD, on the other hand, is an organic disease. You have objective tissue-destroying inflammation.
And you can actually measure that destruction. The sources emphasize that with IBD, doctors see elevated markers in the blood. Things like CRP, which is basically a systemic smoke alarm for inflammation.
A smoke alarm. Yeah, that makes sense.
And they also look at stool markers, like fecal calprotectin. Calprotectin is a protein found inside white blood cells. So if it's showing up in your stool, it means your immune cells are literally bleeding into your gut.
Wow. Which leads to severe risks of structural damage over time, right?
Oh, absolutely.
Things like strictures, where scar tissue builds up from the constant healing and rehealing until it physically narrows the intestine, like it blocks food from passing, or even an increased risk of colon cancer.
Right, that objective damage is the dividing line. With IBD, we can literally see the war zone. And historically, medicine divided this war zone into two distinct territories: Crohn's disease and ulcerative colitis.
Let's map those out structurally. So Crohn's disease is what the sources call transmural.
Yes, transmural, meaning the inflammation doesn't just sit on the surface, it burrows through the entire full thickness of the bowel wall.
Like through the muscle layers and everything.
Through the muscle layers and everything. And it's wildly unpredictable. Crohn's can strike anywhere from the mouth all the way down to the anus.
And it often appears in these patchy areas they call skip lesions, right? Leaving perfectly normal, healthy tissue right in between the deeply inflamed spots.
Exactly. Now, contrast that with ulcerative colitis, or UC. As the name implies, it's confined entirely to the colon and the rectum.
So it never affects the small intestine or the stomach?
Never. And structurally, it doesn't burrow all the way through the wall like Crohn's does. It only affects the innermost lining, the mucosa, and submucosa.
And unlike the patchy, random skip lesions of Crohn's, UC is continuous. It starts at the rectum and spreads evenly upward, almost like a rash.
That's a great way to picture it.
I was looking at the notes on how these two diseases actually got their names, and I mean, the contrast is hilarious. Ulcerative colitis was recognized first, back in 1859, by a British physician named Samuel Wilkes.
Right. He saw a case of bloody diarrhea that just didn't fit the profile of infectious dysentery.
Yeah, and by 1875, they formally established simple ulcerative colitis as a distinct disease, a very clean, logical naming process based on what it is and where it is. Ulcers in the colon.
Very rational, very scientific.
Which brings us to Crohn's disease. And I couldn't believe this when I read it in the sources, but it is the ultimate academic accident.
Oh, it really is.
So it's May 1932. 3 doctors at Mount Sinai Hospital in New York present a paper on what they called regional ileitis. They found 14 patients with chronic inflammation in the lower small intestine, and the doctors presenting were Leon Ginsberg, Gordon Oppenheimer, and Burl Crohn.
A major collaborative breakthrough by all three of them.
Except the journal conventions of the time required authors to be listed alphabetically. C comes before G and O, so Burl Crohn's name goes first. Boom. For the rest of human history, it is Crohn's disease.
Poor Ginsberg and Oppenheimer got completely erased from public consciousness.
Completely. And the kicker is that Crohn reportedly hated having his name attached to a disease that he openly admitted he didn't fully understand yet.
It's a fantastic historical quirk. But what the sources reveal is how our understanding of these two, quote-unquote, separate diseases has fundamentally evolved since 1932.
Right, because they aren't always in neat little boxes.
Exactly. For most of the 20th century, pathologists treated IBD as a strict two-bucket problem. You had Crohn's or you had UC, but nature doesn't care about our neat little buckets. Today, between 10% and 15% of colitis cases show overlapping features that just don't sort cleanly.
So the structural rules break down.
They do. Pathologists now call this IBD unclassified. And because of this gray area, the scientific consensus has really shifted. Well, instead of two completely separate diseases, researchers now view IBD as a continuum of immune-mediated inflammation. Crohn's and UC are just the two most common expressions, the opposite poles of that spectrum.
So how does a perfectly healthy gut slide down that spectrum and turn into a battleground? We have to debunk the popular myth that IBD is simply caused by too much stress or, eating a bad diet.
Oh yeah, it is infinitely more complex than that.
Looking at the pathology in our sources, it honestly sounds less like a disease and more like a riot at an exclusive nightclub.
Walk me through that. I like this analogy.
Okay, so the gut is the nightclub. The intestinal barrier, the physical cells, and the thick mucus layer that is the velvet rope.
Got it.
The immune cells patrolling underneath the lining, those are the bouncers.
Yeah.
And the trillions of normal, everyday gut bacteria, that's the massive crowd out on the street.
What's fascinating here is that your analogy perfectly captures the modern consensus. We now understand IBD as a three-way breakdown between those exact elements.
A breakdown in the relationship.
Right. It's a broken relationship between the barrier, the immune system, and the microbiome.
So what triggers the riot? What is step one?
Step one, the velvet rope drops. The barrier breaks. For decades, scientists debated if IBD was classic autoimmunity.
Like rheumatoid arthritis, where the body just inexplicably decides to attack its own healthy tissue.
Exactly. But in 2001, there was a massive paradigm shift. Two independent research teams discovered mutations in a gene called NOD2.
I saw this. People with 2 mutant copies of this gene have a 20 to 40 times higher risk of developing Crohn's.
It's an enormous genetic red flag. And it matters because the NOD2 gene creates a protein that acts like a sensor for bacterial fragments.
So it's looking for the crowd.
Right. Its job is to recognize normal, friendly bacteria and trigger the production of protective mucus and antimicrobial defenses. It maintains the velvet rope.
So when that gene is mutated,
the gut fails to properly recognize the bacteria on the street. The rope falls down. So it's not strictly autoimmune attacking the self, it's autoinflammatory, a failure to contain the outside world.
The club is suddenly flooded with the crowd. Which brings us to step 2, the immune misfire. Bouncers realize the velvet rope is down, the crowd is rushing in, and they just absolutely lose their minds.
They do.
But if they don't just push the crowd back, they tear apart the club in the process.
That's the tragic part. The innate immune cells activate, and they call in the heavy artillery, the T cells. But the weapons they use differ depending on the disease.
Okay, tell me about that.
Well, in Crohn's, the bouncers mount what's called a Th1 and Ph17 skewed response.
Hold on, let's translate that. What does a Th1 response actually do to the tissue?
Think of T helper cells, like Th1 and Th17, as the tactical commanders. They don't do the damage directly. They release chemical signals called cytokines.
Okay.
In Crohn's, they release specific cytokines like TNF-alpha, IL-12, and IL-23. TNF-alpha is basically an inflammatory flare gun.
A flare gun, I like that.
Yeah, it summons massive amounts of white blood cells that release toxic chemicals meant to kill bacteria, but they end up destroying the intestinal wall itself.
So that's why Crohn's is transmural. The flare guns cause deep structural damage.
Exactly.
And UC has a different tactical commander.
It does. In ulcerative colitis, it's an atypical Th2-like response, relying heavily on a different cytokine called IL-13.
So different chemicals entirely.
Right. And this response is more superficial. which helps explain why UC only damages the innermost mucosal lining and doesn't burrow deep into the muscle.
Okay, so the barrier breaks, the immune system launches all these toxic flares. What is step 3?
Step 3 is the microbial shift. The crowd on the street actually changes its behavior. In IBD, the overall diversity of the gut bacteria plummets.
Like the good guys leave?
Pretty much. Specifically, we consistently see a huge drop in a beneficial bacterium called Faecalibacterium prausnitzi.
That is quite a mouthful. Why does that specific one matter so much?
Because that bacterium acts like the club's repair crew. It ferments dietary fiber into an anti-inflammatory compound called butyrate.
Okay.
And butyrate is the primary food source for your colon cells. Without it, the colon lining literally starves.
Oh, wow.
Yeah, and at the same time that the good bacteria drop, we see a rise in hostile bacteria, specifically adherent invasive E. coli, or AIEC.
So they replace the repair crew.
Exactly. And these strains don't just float by. They actively stick to and penetrate the gut lining, exploiting those NOD2 mutations.
Wait, but if the whole gut is a war zone filled with toxic inflammatory chemicals, how do we know the bacteria actually started the fight? Couldn't they just be the innocent bystanders dying in the crossfire? Like, the good bacteria die, the tough ones survive.
That is the correlation versus causation debate that dominated gastroenterology for years. Are the microbes driving the disease, or are they just a symptom of it?
Right.
The definitive answer came from fecal microbiota transplantation, or FMT.
Oh, transferring donor stool into a sick patient?
Exactly.
Right.
Researchers took stool from healthy donors, processed it, and introduced it into the colons of patients with ulcerative colitis. The landmark trials were led by Paul Moyadady in 2015, and later the FOCS trial in 2017. By completely replacing the microbial crowd with a healthy one, they actually produced clinical remission and intestinal healing in a meaningful subset of patients.
So they just swapped the crowd, and the bouncers calmed down.
That's the takeaway. It proved that manipulating the microbes alone could shift the course of the disease. The microbiome is an active driver of the inflammation, not just a passenger.
But if all this is driven by genetics, like that NOD2 mutation setting the stage, is getting IBD just an inherited inevitability? I mean, if you have the genes, are you just a ticking time bomb?
We know unequivocally that you are not, and that's thanks to twin studies.
Because identical twins share essentially 100% of their DNA.
Exactly. So if genetics were the only factor, a twin of someone with Crohn's would always get it. But they only develop Crohn's about 30 to 50% of the time.
Oh, that's way lower than I would have thought.
And for ulcerative colitis, the concordance rate is even lower, only 10 to 15%.
So the genetics load the gun, but the environment actually pulls the trigger.
The massive gap in those twin studies proves that environmental exposures are decisive. Diet, childhood antibiotic use, infections, they dictate whether the disease ever actually activates.
Here's where it gets really interesting. I was going through the sources on these environmental triggers, and there's one that completely bends the mind. Cigarette smoking.
Ah, the smoking paradox. It is legendary in the field.
It is so incredibly counterintuitive. We all know smoking is a systemic toxin, and if you have Crohn's disease, smoking is a massive risk factor.
A huge risk factor.
It triggers the disease, it makes flare-ups more frequent, and it increases the need for surgery. But, and this is wild, smoking is actually heavily protective against ulcerative colitis.
It's one of the strangest phenomenons in medicine. There were countless documented cases of patients who smoked for years, felt perfectly fine, and then developed severe ulcerative colitis symptoms for the very first time only weeks after they finally quit smoking.
The sources even mention they've studied transdermal nicotine patches as an active therapy for UC.
Yeah, it's wild.
And there's a similar weird quirk with your appendix, isn't there?
The appendectomy paradox. Having your appendix removed early in life, particularly before the age of 20, is associated with a significantly lower risk of developing ulcerative colitis later.
But it increases the risk of Crohn's.
Right. It inversely slightly increases your risk of developing Crohn's.
Which just goes to show, even though we lump them under the IBD umbrella, structurally and mechanistically, Crohn's and UC are operating on very different biological rulebooks.
And deciphering those different rulebooks is what finally allowed medicine to stop using blunt force treatments.
Because for a long time, the only way to treat a flare was to just carpet bomb the immune system with steroids, right? Just shutting down everything.
Basically, yeah.
But given these incredibly distinct pathways, like the TNF alpha flare gun in Crohn's, how did medicine learn to actually fix this precisely?
Well, the watershed moment happened in 1998. The FDA approved a drug called infliximab, sold as Remicade, for Crohn's disease. Infliximab is a biologic drug. It's a lab-engineered monoclonal antibody designed to do one highly specific thing. It binds to and neutralizes that TNF-alpha cytokine.
It just plucks the flare gun right out of the bouncer's hand.
And nothing else. The clinical trials like X and I were revolutionary. Patients who had fistulas and had failed every other therapy suddenly went into deep tissue remission.
That's amazing.
It proved that you didn't have to nuke the entire immune system. Blocking a single chemical pathway can shut down a complex, multi-organ inflammatory disease.
A total game changer.
It really launched the entire modern era of biologic drugs that we now use for everything from rheumatoid arthritis to psoriasis.
But precision medicine hasn't been without its tragedies. The sources detail a famous failure that I think perfectly highlights how terrifyingly complex the immune system is.
Yes.
The drug was natalizumab, or Tisabri, approved for Crohn's in 2008.
The underlying mechanism made perfect sense on paper, too. Natalizumab blocked a receptor called alpha-4 integrin. You can think of integrins as molecular zip codes.
Let's explain that a bit. So white blood cells are cruising down your bloodstream highway. They need to know which exit ramp to take to enter the inflamed tissue.
Right. The integrins act like the exit signs. By blocking the alpha-4 integrin, the drug effectively covered up the exit sign, stopping the aggressive immune cells from leaving the blood and trafficking into the gut.
Which sounds like the perfect traffic control for Crohn's. But alpha-4 integrin wasn't just the exit sign for the gut.
No, it wasn't. It was also the exit sign immune cells used to enter the brain.
Oh, no.
By blocking it systemically, the drug inadvertently locked the immune system out of the central nervous system.
So the brain had no defense.
Right. As a result, a small number of patients developed progressive multifocal leukoencephalopathy, or PML.
A brain infection.
A rare, highly fatal brain infection caused by the reactivation of the JC virus. The JC virus is incredibly common, usually kept in check by our immune system. But without immune surveillance in the brain, the virus just ran rampant.
A completely tragic, unintended consequence of turning off the wrong switch.
If we connect this to the bigger picture, it became a textbook lesson in how translational science has to iterate.
Right, you learn and adjust.
Researchers realized they had the right idea with traffic control, but they targeted a zip code that was too broad. So they went back to the lab and developed a new biologic, vedolizumab.
Okay, how does that one work?
This antibody targets alpha-4 beta-7, which is an integrin exit sign that is completely exclusive to the gut.
So it blocks the immune cells from entering the intestinal fire, but leaves the brain's security system completely intact.
Exactly.
That is incredible biological engineering. But before we look at the global impact, I want to touch on one final, very stark difference in how we handle these two diseases when the drugs fail. And it comes back to the anatomy.
Just surgical divergence.
Because UC is completely limited to the colon, if the disease is just completely unmanageable, a surgeon can perform a colectomy, removing the large intestine entirely. And for UC, that is a physical cure. The susceptible tissue is just gone. But because Crohn's can strike anywhere from your mouth to your anus, removing a damaged section of small intestine doesn't cure you.
No, it doesn't.
The Crohn's can just pop up a few inches down the tract. You can't just cut out Crohn's.
Which is why having a deep bench of these targeted biologic therapies is so vital, and society is going to need them more than ever because the epidemiology of IBD is drastically shifting.
This is the part of the sources that really hits home. IBD isn't just an abstract biological puzzle. It typically strikes people in their teens or young adulthood.
Peak years of your life.
Right when you're trying to finish school, start a career, figure out your life, suddenly you're hit with a chronic disease, frequent hospitalizations, and incredibly expensive medications.
Medications that intentionally suppress your immune system, which brings a whole host of daily anxieties regarding opportunistic infections.
Yeah.
Especially, highlighted during the COVID-19 pandemic.
And it's not just a Western problem anymore either.
No. For a long time, researchers heavily leaned on the hygiene hypothesis. The idea that growing up in heavily sanitized, wealthy environments without enough dirt and microbial exposure causes the immune system to essentially become bored and dysregulated.
Right, because IBD spiked in North America and Europe throughout the 20th century?
Exactly, it fit the historical data. But now, look at the recent work of epidemiologists like Gilad Kaplan. He's documenting a massive, undeniable rise in IBD incidence in rapidly industrializing nations like China, India, and Brazil.
As they adopt Western diets and lifestyles, the disease follows right behind them. So what does this all mean? The sources pointed out that researchers are now aggressively looking at the physical components of our modern diet.
The processed stuff.
Things like ultra-processed foods and chemical emulsifiers.
Emulsifiers are the additives used to keep processed foods stable on a shelf so the oil and water don't separate. The working theory being investigated is that these emulsifiers might literally act like soap on that delicate mucus layer in the gut.
They just wash away the velvet rope.
Yes, directly causing that barrier breakdown and letting the bacteria touch the immune cells. The definitive proof is still being gathered, but the correlation with industrial food systems is impossible to ignore.
We have covered an immense amount of ground today. We started with the alphabetical accident that gave Burl Crohn his famous namesake and mapped out how his transmural patchy disease differs structurally and surgically from the continuous mucosal damage of ulcerative colitis.
And we unpacked the incredible 2001 discovery of the NOD2 gene, which proved that Crohn's begins with a broken barrier, not just an unprovoked immune attack.
We explored the absolute mind bender that is the smoking paradox, proving Crohn's and UC operate on different biological rules. And we tracked the stunning translational science that turned cytokine discoveries into life-changing biologics, learning hard lessons from broad therapies like Tysabri to create ultra-precise traffic controllers like vedolizumab.
It's a testament to how fast medicine can move when it actually understands the underlying mechanisms, the why and the how.
But I want to leave you, the listener, with one final thought to mull over, building on what we just discussed about the global spread of this disease.
Think about this. If the rapid rise of IBD globally is directly tied to our industrialization and our modern food supply, perhaps our highly processed, hyper-sanitized lifestyle isn't just making us generally unhealthy. It might be fundamentally chemically incompatible with the ancient microbial border wall we've relied on for millions of years of human survival.
It's a heavy thought.
It is. If we are structurally dissolving our own internal boundaries just to keep food on a shelf longer, what does that mean for the gut microbiomes of the next generation?
It certainly makes you look at a packaged snack a whole lot differently. Thank you for joining us on this deep dive. Keep questioning the world around you, and especially the world inside you.